Hello everyone, in today's video we are going to be looking at the urea cycle. Urea cycle is made up of two words which are urea and cycle. What is urea? Urea is a nitrogen containing compound which is the primary waste product of protein metabolism in humans and animals. It is excreted from the body through the urine because urea is dangerous to the body. Then, what is cycle? A cycle refers to a series of repetitive processes or biochemical reactions which lead back to the initial starting point. Another important thing which you need to know is that the urea cycle is also called the Krebs ensilate cycle. To make things more easy for you, feel free to grab your pen at this point and write CM. Notice that alphabetically, the letter C comes before the letter M. Now relating this to the urea cycle, C stands for cytoplasm and M stands for mitochondria. Part of the urea cycle takes place in the cytoplasm, while the other part takes place in the mitochondria of liver cells. Imagine this is our liver cell. Let's say here we have our mitochondria, and here we have our cytoplasm. Let's begin with the first step which occurs in the mitochondria. Notice that what really happens here is that ammonium ion combines with CO2. To form this guy here called carbamoyl phosphate. With the help of this special enzyme known as CPS1, also known as carbamoyl phosphate synthetase 1. This free conversion requires 2 molecules of ATP, which convert into 2 ADP. Now we've come to an end of the first step. Notice that for every reaction, we have reactants and final products. The reactants of the first step are CO2, which is carbon dioxide, and ammonium ions, while the final product is carbamoyl phosphate. Notice that still in the mitochondria, ornithine present there waiting for us to continue the process. What really happens at this point? So, let's donate CP as carbamoyl phosphate and O as ornithine. At this point, the reactants are CP, carbamoyl phosphate, and ornithine, and they give the final product which is known as C, also called citrulline. At this point, citrulline will jump back into the cytoplasm. The moment we find citrulline in the cytoplasm, that is the beginning of the formation of adenylosuccinate. But before then, let's take a quick revision on our amino acids. Remember this guy called aspartate with the denotation ASP. This is its molecular formula. The central carbon or the alpha carbon atom, the ordinary hydrogen, the carboxyl group, the amino group, NH3+, the CH2, and then the other carboxyl group. So this makes it negatively charged. Our citrulline combines with aspartate to form this guy known as adenylosuccinate. With the aid of the enzyme known as aginosuccinate synthetase, AGS, with the addition of a molecule of ATP. So, at this point, aginosuccinate is broken down by aginosuccinate lyase to produce arginine and another compound known as fumarate. Fumarate further goes into to join the Krebs cycle. Arginine is hydrolyzed. That means water is added to it to form urea. Let's put it here. And ornithine. Urea is excreted in the urine, while ornithine is transported back into the mitochondria to restart the cycle. That's the urea cycle simplified for you. Each step is vital, and understanding it helps us appreciate how the body handles waste efficiently. Don't forget to check the description for links to additional resources. Thank you for watching.